Okay, so welcome to IB Biology, and right now we're going to be looking at cell respiration, and this particular is just an introduction. I know we've already covered fermentation, but we're going to look at that just one more time and then look at what happens when oxygen is present. So we're going to be focusing on aerobic respiration today. But just as a reminder, here is cell respiration is the carbon cycle, right? So we've got organic molecules that we eat, animals eat, fungi eat, and even plants will take the organic molecules that they made. And what we do with those organic molecules in the food that we eat is we get ATP out of it. And that ATP is used to drive cellular work, like to transport things across a membrane, to move vesicles around. So all that ATP gets used in all the little cellular processes that our body does. It can't take a hamburger and do a cell process with that directly. We need to convert that hamburger into ATP. And then as we do cellular respiration, one of the byproducts is going to be carbon dioxide and water. So you'll see that essentially the equation when we're doing aerobic cell respiration is we have organic molecules like C6H12O6 and oxygen, and what we get out of that is ATP, and carbon dioxide and water are byproducts of the process. Okay, so one of the first things we looked at last time was just the fact that when we take the glucose or the food that we eat, what happens to it is going to depend on whether there's oxygen present or not. But the first step that actually happens will happen whether there's oxygen present or not, and that's called glycolysis. This is the first step in cell respiration. And what glycolysis is doing is it's taking the glucose molecule, which is a 6-carbon sugar, and it's going to split it in half into 2 pyruvates. Each pyruvate is a 3-carbon sugar, so we've gone from a 6-carbon glucose to 2 3-carbon pyruvates. And then if there's oxygen present, of course, it will go into the mitochondria to finish processing, that pyruvate will. If there's not, that pyruvate will then go on to either be ethanol or lactic acid fermentation. Okay. So this is just a quick reminder of the alcohol fermentation. So this is going to happen in bacteria and in bacteria, and some yeast also do this, right? And plants do this. So we've got the glucose molecule breaks down into 2 pyruvates, and then to regenerate the electron carrier that's necessary, the NAD+, to regenerate that electron carrier, we need to use we need that pyruvate to transform into ethanol, and one of the byproducts is CO2. So you need to make sure that you mention that in alcohol fermentation, you start with a glucose, right? And you also, of course, need ADP and P, and you need an NAD+. And what you get out of it is 2 ATP, a couple of these NADH, we're going to look at those carefully. Those are electron carriers. But that pyruvate being transformed into ethanol also releases CO2. So in alcohol fermentation, we release CO2. Whereas when we look at lactic acid fermentation, that pyruvate will be transformed into lactate. And so CO2 is not actually released in that process. So you need to be able to tell the difference. And lactic acid fermentation is what animals do, and there's also some bacteria. And making that lactic acid, if it's bacteria, say, on a milk culture, making yogurt, that's why the yogurt tastes tart, because it's creating more lactic acid. You can actually do an IA experiment watching the pH drop in, when you're making yogurt to see what's happening, because the more fermentation that's happening, the lower the pH will be. Okay, so I'm going to take you through the first step. So remember I said in glycolysis, all right, let's do this here. So I said in glycolysis that we go from a glucose to a pyruvate, and we get ATP out of it, all right? So there's the ATP, and that pyruvate is a 3-carbon sugar. So that pyruvate that's a 3-carbon sugar is ultimately, if there's oxygen available, will go into the mitochondria to finish breaking down. Another byproduct of glycolysis, or it's not necessarily a byproduct, it's a product, it's an important product, is NADH plus H+. This is an electron carrier. So NADH plus H+, this electron carrier, and if you're SL, I think you can just consider it electron carrier. This is going to be really important later on to help make a whole bunch more ATP. When we were doing fermentation, we needed to break that back down into NAD+. But if there's oxygen available, that NADH plus H+ is going to be very important in the 3rd step. Okay, so let's see what happens next. So if there's oxygen, that pyruvate is going to be transferred into the mitochondria. And the next stage is called the citric acid cycle, or it's also known as the Krebs cycle. What's going to happen to that pyruvate is that those 3-carbon sugars are going to, that 3-carbon sugar, each of those carbons are going to be broken down and will ultimately leave as a product of carbon dioxide. So that's where the carbon dioxide comes from that you're breathing out from cellular respiration. Another thing that comes out of the citric acid cycle is more electron carriers. We actually get quite a few electron carriers. In fact, we get NADH plus H+, and we also get another one called FADH2. So for the Krebs cycle, for the citric acid cycle, you take a pyruvate, you break it apart, CO2 comes out. You actually make more ATP out of that because breaking apart that pyruvate provides the energy to make ATP. But it also provides the energy to make more of these electron carriers. So what happens with the electron carriers is that you can see, here's the electron carriers made in both glycolysis and in the Krebs cycle. And those electron carriers are going to be shuttled over to the final stage called oxidative phosphorylation. Those electrons will provide the energy to make a whole ton of ATP, a whole ton compared to how much got made in glycolysis. In fact, if you look at this map here, if you look at this map here on the next one, you can see that glycolysis, you get 2 ATPs. The Krebs cycle, you get 2 ATPs. But that last stage with all of those electron carriers, you end up getting 32 to 34 more ATP. So if there's oxygen available, you get a lot more ATP than if there's not. Because glycolysis is the ATP that gets formed for fermentation, right? So if there's no oxygen, you only get those first 2 ATPs. But if there's oxygen, you're going to get a whole bunch more ATPs available.